Good afternoon, everybody, and thanks, Professor, for introducing me. Today, my topic is rescuing apoptotic neuron from Alzheimer disease using germ, agglutinin, and cardiolipin-conjugated liposomes with encapsulated nerve growth factor and curcumin. Alzheimer disease is the sixth leading cause of death in the United States. Many research have demonstrated that the main reason of Alzheimer disease is the accumulation of beta amyloid in the cortex and the hippocampus. A-beta peptide is derived from amyloid precursor protein, a transmembrane glycoprotein for materials exchange among the neurons and the intercellular matrices. Beta secretase and gamma secretase transform the amyloid precursor protein into A-beta 1,2,40 and then into A-beta 1,2,42. The latter is a strong neuron toxin with a beta sheet structure and appears as non-decomposable neuron fibrily tangled in senile plaques. The accumulation of senile plaques leading the hyperphosphorylation of single tau protein and induced the neuron cell apoptosis on the final stage. DPPC is the major lipid component of the liposomes, and the cholesterol can stabilize the bilayer structure in liposome by increasing in the hardness of bilayer. The segment of polyethylene glycol can protect the liposome from the aggregation of liposome by steric effect. Otherwise, liposome incorporation with polyethylene glycol can protect liposome from the reticular endothelin system and carry the drugs sneaky to the destination. Cardiolipin is an unique amphiphilic lipid with 4 hydrocarbon tails and a hydrophilic head with negatively charged, including the following 2 advantages. First, cardiolipin can be stable the bilayer structure of liposome. Second, cardiolipin favored the ligand binding of liposome to A-beta that can enhance the attachment of liposome to A-beta. Surface modification of germ agglutinin can recognize N-acetylglucosamine, which is generally expressed on human brain vascular endothelin cells surface. Nerve growth factor can regulate the neuronal growth via its 2 major receptors. First, neurotrophic tyrosine kinase receptor type 1, and the second, P75. But the permeability crossing the blood-brain barrier of hydrophilic nerve growth factor is quite low due to the molecular weight and the polarity. Curcumin is an excellent antioxidant and can prevent cyclooxygenases, lipoxygenases, and cysteine oxidase from generating free radicals. But the hydrophobic curcumin with low solubility in plasma can be rapidly metabolized in intestine and the liver. In addition, curcumin with low bioavailability and the stability can be degraded in acidic, basic, and oxidative condition. In this study, we investigate a novel liposomal formulation, incorporation of cardiolipin with germ agglutinin grafting that can enhance the permeability of nerve growth factor and the curcumin across the blood-brain barrier and enhance the attachment of the liposome to A-beta for Alzheimer's disease treatment. As indicated in this figure, an increase in cardiolipin percentage can enlarge the particle size of liposome from 105 nanometer to 165 nanometer, indicating a significant increase in diameter. In addition, incorporation of curcumin and germ agglutinin increased liposomal diameter. This was because the ingredient of cardiolipin and the curcumin might extend the bilayer of liposomal formulation. However, nerve growth factor didn't enlarge the particle size because nerve growth factor was entrapped inside the aqua core of liposome. Thus, nerve growth factor could not affect the surface tension or the bilayer or the liposome size. This table lists the zeta potential of liposomal formulation. As indicated in this table, an increase in cardiolipin percentage can increase in the absolute zeta potential because cardiolipin provides two negatively charged, which is higher than the other phospholipid used. Moreover, incorporation of curcumin and the nerve growth factor didn't significantly alter the zeta potential. This is consistent with the literature because curcumin and the nerve growth factor was entrapped in the bilayer and the aqua core, respectively. Otherwise, grafting with germ agglutinin can neutralize the negative charge on the surface. This was because germ agglutinin carries a positive charge in the grafting medium. Image A to D are SEM image, and we can observe the smooth surface and the enlarged particle size. And this result was consistent with the average diameter data showed before. And the image E to L are TEM image, and we can observe the bilayer structure, which is a typical structure of liposome. And after grafting with germ agglutinin, yield roughly darkness zone overlapping the original bilayer structure. The XPS spectrum shows the intensity of phosphorus in 132 electron volts were maintained at the same level, and the enhancement nitrogen intensity in 401 electron volts when increasing in the concentration of germ agglutinin and the cardiolipin percentage. Table 2 showed the effect of cardiolipin percentage and the germ agglutinin concentration on atomic ratio of nitrogen to phosphorus. As this table indicates, an increase in cardiolipin percentage can enhance in the atomic ratio of phosphorus to nitrogen. And the increasing in germ agglutinin concentration can enhance the atomic ratio of nitrogen. Because cardiolipin contains phosphorus but no nitrogen, and the germ agglutinin carried ample nitrogen that lead the rising in atomic ratio of nitrogen. This table also showed the grafting efficiency of germ agglutinin on liposomal formulation. As this table indicates, an increasing in cardiolipin percentage can enhance the grafted efficiency of germ agglutinin, because germ agglutinin with positive charge in grafting medium can be attracted by negatively charged cardiolipin in liposomal bilayer via electrostatic interaction. Otherwise, self-competition of germ agglutinin leads to the decrease in grafting efficiency of germ agglutinin. In this figure, an increasing in cardiolipin percentage can enhance the entrapment efficiency of nerve growth factor and the curcumin, because the hydrophobic interaction between cardiolipin and the curcumin causes cardiolipin easily to fasten curcumin in the bilayer. And the hydrophobic head of cardiolipin with negative charge can attract the positive charge nerve growth factor and catch the nerve growth factor inside the aqua core. And in figure A, liposome incorporation with curcumin can reduce the entrapment efficiency of nerve growth factor, because the interaction between curcumin and the membrane lipid might extend the bilayer region and shrink the core space in the liposome. In figure B, incorporation of nerve growth factor can enhance the entrapment efficiency of curcumin, because positively charged nerve growth factor merged uniformly near the inner bilayer, leading to an uniform distribution of negatively charged phospholipid in the liposome and accelerating the curcumin encapsulation rate. In this figure, an increasing in cardiolipin percentage can slow the release of curcumin and accelerate the release of nerve growth factor from liposome, because of the strong affinity of cardiolipin for curcumin. Otherwise, high level of cardiolipin reduce the cholesterol content in the lipid, decrease the liposome tightness, making nerve growth factor easy to migration from the liposome. Otherwise, the accumulated percentage of nerve growth factor released from liposome was less than curcumin released from liposome in early 6 hours. And the accumulated percentage of nerve growth factor and curcumin were comparable in 48 hours. This figure showed the viability of HB max, human astrocytes, human brain vascular pericyte, and the SKN-MG cells stimulation by by liposomal formulation and without A-beta induction. The viability data demonstrate liposome with germ agglutinin grafting can slightly increasing in the cytotoxicity to the blood-brain cells and the SKN-MG cells without A-beta stimulation. Because the positive charge of germ agglutinin could trigger electric intervention in the negatively charged cell system. Moreover, no significant difference in viability of HB-max, human astrocytes, human brain vascular pericyte, and SKN-MC cells was found between bare liposomes and curcumin liposome formulation. This suggested that the current liposome could avoid a strong cytotoxicity induced by curcumin. In addition, there is well known that the accumulation of lipid in endothelial cells can induce apoptosis because the oxidation products of lipid, such as 7-asterol, is damaged to endothelial cells. However, the cytotoxicity of liposomal formulation to the blood-brain cells and the SKN-MC cells was quite low and satisfactory for drug delivery system. The permeability can prove the uptake of liposome by HB-max and the human astrocytes. The permeability of nerve growth factor and the curcumin did not significantly enhance when increasing in cardiolipin percentage. But after grafting with Huijum agglutinin can improve the permeability of nerve growth factor and the curcumin across the blood-brain barrier. The slight permeation of free nerve growth factor crossing the blood-brain barrier and the HB-max and the human astrocyte cells result from its high affinity for neuron-trophic tyrosine kinase receptor type 1 expressed on HB-max. In addition, HB-max carry the negative charge can attract the positive charge Huijum agglutinin while electrostatic interaction and activate adsorptive mediate transcytosis. Moreover, HB-max carry an N-acetylglucosamine, which is recognized by surface Huijum agglutinin. Even though the permeability of curcumin liposomal formulation is quite higher than free curcumin, but they can reduce the toxicity and enhance the bioavailability of curcumin. With different concentration of A-beta, the viability of SKN-MC cells decreased with time and the higher concentration of A-beta. Treating with liposomal formulation for 24 hours and 36 hours significantly enhanced the viability of SKN-MC cells. Because the cardiolipin has strong affinity for A-beta fiber and the oligomers, and the curcumin can inhibit the aggregation of A-beta and the destroy of A-beta fibers. Moreover, SKN-MC cells can be responsive to nerve growth factor due to the expression of neuron-trophic tyrosine kinase receptor type 1. Thank you. Which is the major receptor of nerve growth factor. This image showed the uptake of liposomal formulation by HB-max. The green intensity in image A to D were maintained at the same level when increasing in cardiolipin percentage. Liposome with Huijum agglutinin grafting can enhance the uptake of liposomal formulation by HB-max, and we can observe the enhanced green intensity in image E to H. And the green intensity was enhanced when increasing in germ agglutinin concentration. And this result was consistent with the permeability data showed before. The uptake of liposome by SKMC cells induction with A-beta can be observed the enhanced green intensity in image C to E when increasing in cardiolipin more percentage. But the green intensity in image F and G were a little weaker than image C to E. This was because surface germ agglutinin could slightly hinder the diffusion, hinder the attachment of liposome formulation to A-beta near SKMC cells. And the conclusion are, first, liposomal formulation are low cytotoxicity and satisfactory for drug delivery system. Second, liposome formulation with germ agglutinin grafting can target blood-brain barrier and enhance the permeability of nerve growth factor and the curcumin crossing the blood-brain barrier, and enhance the bioavailability of curcumin. Third, cardiolipin can enhance the attachment of liposomal formulation to A-beta and protect SKMC cells against neuron toxicity. And thank you for attention.
Electron microscopic features of the liposomes. You somehow measured the size, and I was wondering how did you do that, and how many liposomes did you measure? I just have trouble imagining like measuring a size from a 3D object in 2D perspective and getting enough data so that the conclusion would be statistically relevant.
The liposome diameter we observed by DLS, and the total.
The next picture, the next slide. Where you have the 10 images. The electron microscopy pictures. Can you just go one step further or two?
The SEMT image, SEM,
Yeah.
or TM.
Yeah, that one. Perfect. How did you size these particles? How did you say that the particles get bigger or smaller? Or did I get something wrong?
And SEMTM, the image showed the particle size is always smaller than the DLS measurement. Because the now SEMTM we must to dry the liposome and in the vacuum.
Oh, you mean this comes from
So the liposome will be shrink, be small, and then in water, in the medium or in the buffer. The buffer liposome in the buffer is larger than dry liposome will be small.
Did you test the effect of liposomes and these conjugates on apoptosis?
Apoptosis.
So the answer is no. No induction of apoptosis. That's
Yeah. We measure the viability is not satisfactory. Not is over 80% for these cells, and we
This figure shows that there is no effect on proliferation. That's what you mean.
yeah, but I think that if the time, the viability will be different when the time for a long time. But at least we test is for 8 hours to 60 hours.